Hello, welcome to the first lecture from biological chemistry, enzymes, during which we will discover classification, nomenclature, characteristic, regulation of enzyme activity, and using of enzymes in pharmacy and medicine. In 1964, the International Organization for Biochemistry and Molecular Biology divided enzymes into classes, each with its own number. The name of the class indicates the type of chemical reaction that is catalyzed by the enzyme. Class number 1, oxidoreductases. Second class, transferases. Third class, hydrolases. Fourth, liases. Fifth, isomerases. Sixth, ligases or synthetases. And seventh class, translocases. The classification system provides a special code for each enzyme, consisting of 4 code numbers separated by dots, and specifies the action of the enzyme on the chemical group of the substrate, the type of chemical bond affected, etc. All enzymes are coded only by International Enzyme Committee. There are few types of nomenclature unknown: trivial, formed historically, working nomenclature, and systematic. On this slide, you can see three-dimensional structure of some of the well-known enzymes: alcohol dehydrogenase, ATP synthase, alpha amylase, and catalase. Enzymes have all the features of the structural organization of proteins. Like other functional proteins, enzymes are divided into simple enzymes, which contain only the protein part, and complex enzymes, which are containing protein part, apoenzyme, and non-protein part, coenzyme. Coenzyme usually presented by vitamins, nucleotides, metal ions, etc. In three-dimensional structure of a simple and complex enzyme, areas with certain functions are defined. So active site of enzyme is a place in the structure of the enzyme molecule in which the interaction with the substrate occurs. Usually active site is formed by amino acid residues of the polypeptide chain of the enzyme molecule that you can see on the example of chymotrypsin. Substrate is a substance that is affected by the enzyme during enzymatic catalysis. For example, the substrate for chymotrypsin are proteins from food, specifically the peptide bond in their structure. Enzyme-substrate complex is a complex in which the enzyme and the substrate are binded to each other by chemical bonds. In the process of enzymatic catalysis, enzyme-substrate complex is formed during successful catalytic process. On the slide, you can see the polypeptide in the active site of chymotrypsin. After a successful catalytic process, the product is formed. As an example, enzyme chymotrypsin breaks down proteins into individual amino acids. All catalysts, both biological and non-biological, follow to the general laws of catalysis and have the following similar characteristics. Catalyzes only energetically possible reaction. The reaction rate depends on the energy barrier that needs to be overcome by a reactant. If activation energy is high, the higher barrier needs to be overcome that take and make the rate of reaction slow. Catalysts decrease the barrier, resulting in an increase in the number of reactive molecules, thus increasing the reaction rate. Both protein and non-protein catalysts do not change the balance of the reversible reaction, but only accelerate its completion. Catalyst never changes the direction of the reaction and are not consumed in the reaction process. However, enzymatic catalysis has features which differ them from non-biological catalysis. The rate of enzymatic catalysis is much higher than non-enzymatic. Dependence of the enzymatic rate on the substrate concentration is described by Michaelis-Menten mathematical equation, which you can see on the slide. Enzymes have high specificity. Specificity is the affinity of the enzyme to the substrate. Reactions are going in mild conditions. Normal pressure, low temperature, and pH of the environment, which close to the neutral and correspond to the environment inside the organism. And the rate of enzymatic reaction is directly proportional to the amount of enzyme. Enzymes are catalysts with regulated activity. Activators is compounds that accelerate the enzymatic reaction. On the slide, you can see the example of activator. Inhibitors are compounds that slow down the enzymatic reaction. Competitive inhibitors is similar in the structure to the substrate and binds to the active site. As an example, a drug allopurinol is competitive inhibitor to enzyme xanthine oxidase. Non-competitive inhibitors bind to the enzyme inside or outside the active site, preventing interaction with the substrate. As an example, you can see on slide, cyanides inhibit enzyme cytochrome oxidase. Allosteric modulators interact with the enzyme in the allosteric site, outside the active site. Several mechanisms are important in the regulation of enzymatic processes. Changing in the number of enzyme molecules, availability of substrate and enzyme molecules, and change in the catalytic activity of the enzyme molecule. Adaptive enzymes are enzymes which synthesis begins in the cell only with the appearance of specific modulator. Genes that control the biosynthesis of adaptive enzymes usually exist in the repressive condition and are activated only in the presence of inductor. Also, some medications, such as barbiturates, can provoke metabolic tolerance to drug by induction of microsomal enzymes in liver. Catalysis activity can be regulated by controlling the income of substrates inside the cell and removal of products or intermediates outside. This mechanism includes diffusion, active transport, etc. In a eukaryotic cell, most organelles are compartments. Each of them is characterized by a certain list of enzymes and catalyzes by them metabolic reactions. Enzyme activity might be controlled by chemical modification, such as phosphorylation and dephosphorylation reactions. This process is carried out by specific enzymes, protein kinase and protein phosphatase, the level and activity of which is regulated by hormones. One more way of changing enzyme catalytic activity includes interaction with modulatory proteins such as G protein. The mechanism of self-regulation of enzymatic processes is based on autocatalytic reaction. It includes transformation of proenzyme or non-active enzyme into active form by removal of some part in polypeptide chain. Isoenzyme is the various molecular forms of one enzyme, which is different in electrophoretic mobility. Isoenzymes catalyze the same reaction but in different organs. Well-known example of isoenzyme is lactate dehydrogenase, LDH. Each organ is characterized by a clearly defined isoenzymatic spectrum. For example, in the heart, localized isoform 1, LDH1, which contains 4 H subunits from H heart, while in muscle, isoform 5 prevalent. Isoform 5 contains 4 M subunits from M muscle. Isoenzymes have great diagnostic value. Knowledge about enzymes are used in medicine, in particular in the prevention, diagnosis, and treatment of disease. So there are 3 main fields in enzymology are known: enzymopathology, enzymodiagnosis, and enzymotherapy. It is believed that the development of the disease is most often associated with hereditary insufficiency of complete lack of synthesis of a single enzyme in the patient's body. The purpose of enzymopathological research is to solve problems related to pathogenesis and molecular mechanism of replacement of violated enzyme function. The purpose of enzyme diagnosis is to establish a diagnosis by determination of enzyme activity in biological fluids of the body. In case of cell damage during pathology in the blood, increases the concentration of intracellular enzymes, which can be detected. The idea of using of enzymes as medicines has always seemed attractive. Researches are conducted to find potential remedies for the treatment of tumors, cardiovascular diseases, etc., among enzymes or agents that are able to regulate enzyme activity. The mechanism of action of already known drugs we will study during the course biological chemistry. So please remember that enzyme name has an ending "ase", and drugs regulate the activity of enzymes by competition with the substrate for the active center of enzyme and chemical modification of the enzyme molecule, etc. Thank you for your attention.